Assalamu alaikum all. Hope you all will be doing good and you have a very good learning experience for the yesterday class. So today we are going to continue with the virtual screening process. So lastly, we have prepared the PDB-QT file formats of the ligand and the protein themselves. And the most important thing which we will be discussing today, which halfly we have done the work yesterday, is the preparation of the configuration file. So this configuration file is actually contain the name of the receptor and the centers and the size and the number of modes which we have to run. So this configuration file should be within the folder of the where you have to run the virtual screening. That is the protein PDBQT and the PDBQT formats of your ligands, which we are going to screen. This center 2, 6, and 7 of the XYZs has been determined, which we have done lastly by the active site predictions. So when we get the amino acids and the values of the XYZ coordinates, they are going to be put over here. So the utilization of learning with how we can identify the amino acids which are involved in receptor binding affinity and their attributes, they are going to be utilized. So this size of the grid box is specific, or we try to make it not too big as it covers this sequence of amino acids and the number of modes should be 9 averagely. It should not be more than 15. It should not be less than 8 so that we can have the different poses or the different models of the screened molecules. So this configuration file is an important member and it has been determined by preparing the by identifying the amino acid. As in both the cases, like we have previously discussed, I have given you some articles where the configuration XYZ coordinates has been given. So if you are working, just like I have given you the activity to perform, you are going to put the name of the receptor over here, which you are going to convert into the PDBQT format and the XYZ coordinates over here. And then you will keep the same and run within the molecules, and you will get the virtual screening done against that specific receptor protein. But in some cases, you are not getting it through the literature survey. You have to find it by yourself. For that, you can use the PDB sum and PatchDog, and there are so many other web-based servers which you can do. So this configuration file is prepared. We make sure that this configuration, the name of the receptor, which has been written over here, that is the one karen clean underscore h, should be the same name as of your PDBQT format of the receptor protein. Otherwise, it is not going to run. And in this text file, we have also mentioned the name of the receptor, number of modes. It should make sure that every ever since some other time if you are going to perform the same molecular docking and the virtual screening, use the same text file or make the changes just within the receptor and all those, because these files, these configuration text files and the shell script files are actually the one which are case specific, and even they take the extra space as their evaluator, or in other words, for example, if I make this space less, so it is going to disturb the identification of the molecule. So it has to be used as such. So every time you can change the name of the file, but you should not change this space between the is equal to. So what you should do, you just make the change over here and other thing. Okay. So don't make any other changes, and you can use this text or the notepad file for the preparation or the fun as the configuration file for any other protein, but do change the things which need to be changed, otherwise keep it as such. Secondly, we also have this Vina underscore shell script file. This shell script file is the one which is responsible in the working of your Vina. So what we have done over here, that this we have mentioned the name of the ligands, okay, in the PDBQT format. We have mentioned the pathway and we have mentioned the output file. That is, okay, in this script, we have the information of the input file, where these input files are there, where this configuration file is there, and the output file. We can change the output .pdbqt because it is not working as an input of any system. Output है कुछ भी निकल के आएगा. It doesn't need to be the same. Cannot be need to be mentioned. We just said कि out.pdbqt. But the input file system or the parameters where they have been placed, that is, they are saying it is placed in the C Cygwin, although right now we don't have it in the C Cygwin. So what should we have to do? We have, which means okay, this shell script is going to work when all these parameters or all this folder is actually in the C Cygwin endpoint. Okay. First point, along with that, we only need the PDBQT format of our ligands of our receptor protein configuration vina.exe. So we just have the information that all these things are present at the C Cygwin home vina test. Cygwin I have already provided you, which is required as a simulation environment for the Linux system with the Windows. So that is why we are going to work over it. So firstly, what we have to do? We just have to, along with that, there's one another change here. Being a test, we don't have to hit the folder is not the being a test. Okay, it's a workshop activity. So we have to change, or we can only need to change these things. This, we don't need to change this, not this. So we just have to put this folder at the C Cygwin home, and either that folder name is Vina test or whatever you want to put. So what I have done, I have copied it, and then I went to the C folder. Okay, C Cygwin home and put it over here. So when I paste it over there, in order to change it, I have done it by the name of the Vina W. So the file, the folder which I have shared with you, activity 2, contains all the PDBQT formats, and the folder which I have again shared with you with the name of the analysis, that is the one where the execution has been done. Okay, so these are all the output files which I have already run. So again, in order to check or to identify that how we can do it, so I am going to open the C Cygwin. Okay, again, C Cygwin home. Okay, and I'm going to place it over here, that activity 2. Okay, so the location is now C Cygwin home, and the folder which we need to run it or where we have to run the activity is the C Cygwin 64 home activity B. So firstly, what we have to make change in the configuration file? No, it's all set to go. We just need to change in the Vina shell script. So what we have to do? We just have to copy it. We don't need the spaces over here. So I have changed it over here and again renamed it by changing the space. Okay. Okay. The configuration file shell script I open. So activity 2, change the Vina shell script segment 64 home Vina test. And the location here is this one. Now, just note down one thing that the slashes over here are in the different direction. Okay. While here we have the backslashes. So when we are going to put this, we have to change the slashes. Okay. And we don't need to make any other change. And I have saved it. Okay. Now we have to go to the Cygwin terminal. So I'm going to write Cygwin. It gives me this. Okay. And then we have to go for the PWD, define the pathway, LS, okay, CD space, home, Okay, home. LS. It's L for loin, not one. Space. Okay, need to paste it. Go to the terminal. Okay, it's not working right now. I'll make it check. So, what we have to do firstly, we have to identify where we are, define the pathway. The same que we are at this position, change directory, cd space dot dot enter. So it has changed the pathway to the home page. Now in home, what we have to do, ls, locating the folders, activity do. Now we have to go to the folder activity do. So cd space activity to enter. So now as the pathway has been defined, what we are saying is the home activity 2. We are here into the folder where activity 2 is there. So here we are going to write the script sh space Veena underscore screen underscore local dot sh. Okay. And when we paste the enter, the processing is going to be start. There is change in the folder. Okay, the screening is in process and the virtual screening has been started. So by the end of this virtual screening process, what we will be having is that the sequence has been or the table has been generated. So the ligand one molecule has been completed. It is telling you that this part has been completed, and we are getting the RNSD, the binding affinities of each and every molecule towards the one single protein. So as it depends upon your processor type and the configuration with how much time it is going to take. So as we have a smaller number of molecular groups, so it's not taking that much time along with the configuration of your system also plays an important role with how much time it is going to take. So now the virtual screening of all the molecules has been completed over here, and what we will find that these folders has been generated. So these are the folders where the results of your protein of your systems are there. So you will have the log.txt file, output.pdbqt file, which we are going to do the analyze during our sessions, like in second part of the session.